Good afternoon. I'm Esther Maina, Department of Biochemistry, and I think I will start us off by giving an introduction into genomics, and more specifically, I'll be talking about the African genome, the reasons why we need to study it, what are the advances in technology that we are experiencing at the moment, which actually strengthen our fight or our cause towards understanding our genome, and what is the end story. And some of the work that the group that I work with is involved in. So I will, okay, thank you very much. So again, like I said, I'll be talking about the African genome, and more so for this session, it will be quite introductory and actually just trying to drive the issue or the agenda of normalizing this narrative in the African scientific fraternity. So I've actually termed my talk The Long Walk Home, case in point being Africa is the cradle of mankind. So yeah, so I will begin. So a genome is a complete set of genetic information of an organism. It actually includes all your genes and the proteins, which actually subsequently give us the functions therein. So the bringing together of all these moieties actually are what give us a genome. So we'll look at that and why we need to study it, and some evidences as to why the African genome needs to be given special attention, some evidences, and then we'll take it from there. So this is a quotation by Charles Rotimi, the gentleman from NIH who started off the H3 project or consortium, which kicked off the studying of the African genome, where he says that there are things in the human genome that we cannot study anywhere else but on the African context. So he goes on to strengthen my argument that Africa is the cradle of mankind. Therein lies all the basic and the originals of genomic information that we so seek. So why do we need to study the genomic sphere of Africa? So genome research actually is search for variation. That is how I see it, and other researchers. Roughly, we do know that at least 99.9% of our genomes across populations are quite similar, but what remains different, or the difference therein, is what is important. This is what we are seeking after, because it will give us answers that lead us to actually determine what causes our diseases, both communicable and non-communicable. I'm a non-communicable disease researcher, specifically cancer. So finding those differences will go a long way to enable us determine or interrogate the billions of nucleotides in our genome towards that end. The lack of African genetic material, so we do not have biobanks because we do not have those genetic materials. This constitutes significantly an obstacle, or poses as an obstacle to our understanding of how our bodies and diseases function. Case in point is the narrative of cancers in Africa, and I'm sure many of you have encountered patients who actually show very poor response to chemotherapeutic agents. We do not understand the mechanism of disease because they are not actually working together with what is in data at the moment from the Caucasians, which is the other population. So Europe actually constitutes 16% of global population. But if you look at the genomic information, or the DNA information that is out there, it constitutes from Europe. It is quite, Africans actually are actually presenting some dismal presentation, because we only present 2%. That is how little we participate in terms of these genetic studies, and that is to our detriment. Means that our populations are significantly and severely underrepresented. And so in the event that we wanted to find these differences that I so daily talk about, issues of comparative studies of genotype, phenotype correlation studies are not possible at all. So it is important to actually push forward the issue of understanding the human genome so that we'd be able to do comparative studies of our diverse population and understand the genetic basis of the phenotype, adaptation, and complex diseases that we see in all our populations. So Africa actually hosts the majority of human genetic diversity, which we'll see shortly. Obviously, it holds the clues to human origin, it's the cradle of mankind, and unfortunately carrying the heaviest disease burden because of our diversity. Yet genomics has so far focused on our European counterparts, but now Africa and other populations are looking forward or hoping with the advancement of research and techniques and technology to reap the benefits as well. So globally, human populations show structured genetic diversity, and this is as a result of geographical dispersion, selection index, what pressures are put on us, and so it even affects matters of drifting. These variations subsequently will provide insights into the evolutionary processes that shape both human adaptation and variation in disease susceptibility, not only to the Africans, but to the human race, so to speak. There are two projects that have greatly enhanced our understanding of genetic variation, and they're sort of early days. The Hackman and the 1,000 Genome projects, which have been quite advanced and enhanced our understanding. But the characterization of African population remains limited or wanting, so to speak. So this is the first, I've selected work by this scientist, Gardasini. She's done a lot of work, and I've started with some of the work that she's done nearer to home, which is work that they did in Uganda, looking at the population history and genomic discovery. That work was done in Uganda, and it really was shocking. This was work done in 2019. The first report was that more than a quarter of the genetic variation that were found among the people of 25 Ugandan villages has never been recorded previously. So that is just Uganda. We're talking about 25 little villages. And so in the grand picture, and the grand scale of issues, if you were to think of Africa in its entirety and the diversity of the people therein, then one must then be able to project and see that we're actually quite wanting in this. And that is why, again, I will say that this narrative has to be pushed forward. Then after that, Gradasini et al., or actually it was not after, these are two things that that group has done. They had actually started looking at the Sub-Saharan Africa, looking at the genome variation under the African Genome Variation Project, which is an international collaborative effort that has actually expanded the efforts to systematically study and assess diversity among 1,481 individuals from 18 ethno-linguistic groups. And you will be happy to know that Kenya was involved in this one. We had the Kikuyus and the Kalenjins were actually captured. It's just sad that it is not a Kenyan initiative or an advancement as we see in Uganda, because the Ugandans have actually taken the first step to start studying their genome. So actually, they actually got some very interesting results, which I will share shortly. So they used an analysis from all this diverse work. So out of the 1480 individuals, they used 320 to do a whole genome sequencing analysis, which is quite a lot, using the next generation sequencer. So you can imagine to do the whole genome. It was quite impressive. using the admixture analysis, which is one that shows how the substructures within populations actually interact, thereby giving us diversity. How that is what this statistic in the context of Eurasia and the African context, they were able to then show us what is the output of the same. So I will share so that you have an appreciation. You can see that in this presentation from the admixture analysis, you can see that the Europe comes in as a block, Asia as a block, the Americas, and then Africa is actually subdivided. When you look at North Africa, it has substructures. These are all genetic diversities, the variants that were reported from this. That should then tell you the amount of data that is wanting, the variability therein that will go a long way to advise us in issues disease, in issues, for example, in my area, in issues cancers, so that as I ask questions about prostate cancer and breast cancer, then I know that there's a lot of variation, even in my own population. So I thought that was quite interesting. So as a summary, they actually presented 2.2 million modest variants that show differentiation among the sub-Saharan Africans. They did report 29.8 million SNPs, single nucleotide polymorphisms, that were identified from Ethiopia. And in Ethiopia, there are some substructures of the Oromo and the other small tribes therein, the Zulu, the Baganda. So that was work done on the whole genome sequence by the ABGP group. So I hope you now have that understanding and appreciation of the diversity that we have that has not really been tapped in, and that we need to harness our resources and our collaborative efforts to that end. So to conclude the AGVP work, I would then say that African populations are characterized by greater level of genetic diversity, extensive population substructures, as we've seen from the admixture analysis, and so less linkage disequilibrium compared to the non-African populations. Africans also possess a number of genetic adaptations that have evolved in response to diverse climates and diets, the nutrition, because we're very diverse as people of many colors and many traditions and many things, as well as exposure to infectious diseases, which we're obviously very well aware of and familiar. So at least this AGVP result from 2014, 2019 actually should strengthen our wanting to delve into the African genome the more. So in terms of the technologies and the advancements therein, because this is where our salvation comes from, there's been unprecedented advances in molecular and cellular biological tools. For example, the cost of sequencing. There are times when, I remember up to 10 years ago, sequencing was something that could not be discussed in Africa, but up to yesterday, you can actually sequence a primer pair for 1,000 shillings. So you can even fund some of your sequencing work. And so those drivers of innovation in chemistry, optics, fluidics, computational bioinformatic tools, driving the costings of these technologies or biological tools to an affordable level should strengthen us the more to want to get involved in this. So that has actually been very important. And indeed, we're moving from the dideoxy sequencing to now the next generation sequencing, where we can now look and interrogate whole genomes in their entirety at prices which we can now term as affordable. So we are in the multi-omics era, which is quite interesting, and And I would probably say we haven't heard of others. We are working, and when I say we, I work in a group of some other researchers from the University of Nairobi and Manchester University to use multi-omic approach to ask specific questions about specific manifestations, and specifically we are looking at prostate cancer. And so we have sections where we are doing the genomics, proteomics, which is looking at structure and function of proteins, transcriptomics, which deals with matters RNA. So we're interested in gene expression and the sequence of the transcriptome. Metabolomics, where we assay for metabolites that come from a situation or a disease, and looking at how that affects that specific manifestation or pathophysiology that we are observing. You know the roles of microbiomes in terms of, for example, in the gut and how the immune system is regulated by that. So basically, we are doing a multi-omic approach towards prostate cancer as our starter point. So towards that end, we will use some statistical analysis, which actually has factors or linker factors that actually give us a weighting to each of the omic towards how much it builds towards a specific manifestation. And like I said, in my area of prostate cancer, we'll be looking hypoxia that is created in cancerous situations, and look at the metabolites therein and see how much they actually impact on the disease severity or progression, and why we see abnormal, and by abnormal meaning that it deviates from what is in what is published data. So in terms of talking about multi-omic approach, this is what would be able to ascertain, be able to carry out patient stratification, predictions of therapeutic responses, personalized treatment, most important, the therapeutic monitoring, going towards prognosis. So this is bringing together of those omics. So what are the gains from these advances? All these developments define this, all the knowledge of us wanting to challenge the African genome, and then also being supported by advances in tools and what the molecular biology techniques. We should be able to, we are actually in a unique and opportune time where we can seek to understand our African genome. We can now envision obtaining easy access to the complete sequence of 3 billion nucleotides. We can interrogate them further and do our mechanism of action results, and even trying to understand signaling pathways of specific disease, subsequently leading us to genotype-phenotype correlation analysis between our population and populations out there. This knowledge that we will gain from such a venture will have a major impact on healthcare, disease progression or prevention or management, to also raise challenging issues regarding rational issues and ethical implications, which are also very important. So what are the ongoing things or research work in Africa? And I've actually singled out two major consortiums. The one that was founded by Charles Rotimi, which is a H3 Africa. And you can see it's actually with collaboration with Wellcome Trust. They actually have quite a diverse sort of like collaborative efforts across Africa. And this is with a view of covering the whole of Africa, to actually look for the diversity in our genome and the African, and actually creating databases from where we can pull out our genomic information and be able to do subsequent work therein. So H3 Africa It's an initiative that consists of 51 African projects that actually deal with both communicable and non-communicable diseases in Africa. What really impresses me about H3 is how it empowers the African researcher in genome science by enhancing or strengthening collaborative efforts, and again, the facilitation of the fundamental research work is actually led by African scientists for the African people. So H3 Africa has gone a long way in issues of creating or strengthening capacity, which is much needed in our background. The other one is 54 Gene, an initiative which is focusing on all the 54 countries in Africa. It's Nigerian-based, and it was started by Dr. Bon. Basically, they're actually dealing with drug creation or drug discovery. And this actually is very important because we have the H3 Africa doing the genetic diversity studies, trying to understand the African genome. And then we have this other arm of 54 Gene trying to do drug creation. So, and both of these situations run concurrently, which is exactly what Africa needs now. So what are some of the challenges? The challenges are just the usual challenges that you would have. Of course, there will be some that are a bit odd because of the kind of data that one is dealing with. For example, discovery of incidental findings unrelated to disease in question. Patient awareness in our population is quite difficult. That's the usual. And you will note that I have not mentioned finance, which is actually a global issue. It's an issue that affects all researchers globally. Of importance to me and fellow researchers is who then owns the data. That is a question that we then must ask, because we still are resource constrained in terms of infrastructure. And so we rely heavily on our collaborators to do the slightly more complicated analysis out there. And so we send out our samples. So this is some discussion that has to be ongoing so that we know who owns the data therein. There definitely will be some good opportunities. I'm sure you've seen that in terms of precision medicine, where we're able to personalize treatment that will come from understanding of the genes and be able to use that information to actually tailor-make treatment. A good example is what was done 20 years ago by Golub et al. Matters leukemia, where Golub et al. Came up with a subset of 6 genes that could actually then break down leukemias and be able to very quickly determine the exact leukemia that a patient is presenting, subsequently quickening the treatment protocol, and it is quite precise. And that is where we are actually looking to go. And in as much as it is a lot and it's quite demanding, I don't think it's insurmountable. I would not want to finish this work without talking about novel biomarkers. That has actually been my absolute interest in matters, cancers in Africa, because I'm very sort of pushed by matters of how diseases are progressing, the severity, the prognosis. If we're able to identify subsets of markers that are unique to our populations, for example, in breast cancer, we'll go a long way in understanding these diseases and being able to understand the molecular biology therein. Other issues are issues of costs. Can we come up with cost-effective designs for carrying out genome-wide association studies in Africa? Because you want to then, once you've pulled out the biomarkers, associate them with a specific phenotype, and so have an ending to a story, thereby leading you to a way of sorting out or mitigating the situation. Of course, the other good things that will come out of all of this is a collaboration across the regions, strengthening research capacity. We envisage data from these projects will provide a global resource for researchers, as well as facilitate genetic studies in Africa. Of course, research infrastructure overall improvement in disease management. So I would like to acknowledge the team that I work with. These are the people involved in the multi-omics team, and hoping that the next meeting or the next invite I get from this background, we will be having some very interesting results from our prostate cancer. So there's Dr. Ezekiel Nature, Dr. David Wedge, Professor Peter Mungai, Dr. Esther Kanduma, Dr. Martin Mlinge, and Professor Robert Bristow. And for me, Esther Maina. Thank you very much.